Hi everyone. In today's video, we'll be going over Lab 1, Solutions, Measurements, and Microscopy. I want to emphasize that today's lab is particularly important as you'll be using the concepts and techniques you learn in this lab in future labs. In Lab 1, you will create a buffered solution and perform a few serial dilutions. You will also count beads using a hemocytometer, as well as use the plate reader, a pH meter, and microscopes. Because you will be using pipettes and pipette aids extensively in this lab, it's important that you learn to use them properly. Be sure to read the posted articles, as well as pay attention to when your TA is demonstrating these tools in your lab. I just have a few tips for you guys. In particular, I think it's important that when you first start using these tools, to use them slowly. And what I mean by that is when you are either pressing or depressing the plunger, be sure to do so slowly. In addition, when you're first getting your pipette tip wet, you want to expel and draw up the solution a few times, as this will help you to eject a constant volume. Before we move on to the procedure, I just want to point out that you should be looking at the MSDSs for the different chemicals we're using in this lab. In particular, we're using hydrochloric acid and sodium hydroxide, both at a concentration of 0.5 molarity. This is not particularly strong, however, you still may experience discomfort if you spill some. So read up on the safety information so you're prepared in case that happens. In the procedure today, you will be weighing out a few solids as well as adding liquids to them. It's not particularly difficult, but I do want to point out that 120 milligrams as well as 25 milligrams is actually a really small amount. So when you're first adding these powders to the scales, make sure you add only a little bit at a time. It's very easy to add more powder to the scales than it is to remove it. In addition, getting exactly 120 and exactly 25 milligrams is not particularly important. So if you end up adding a little more, a little less, don't worry about it. Just make sure you record it in your lab notebook. In this lab, it's not particularly important to get the exact amount, but in the future, some of your reagents may be a lot more expensive, so you'll want to be a lot more careful when you're measuring those. In the second part of the lab, you will be using the buffered solutions you create in the first part of the lab to generate a serial dilution. This is meant as practice, so please have both you and your partner do this. For example, because you want to have 200 microliters in each well at the end, what you're going to want to do is something like add 400 microliters of your solution into well 1. Into the other wells, so in this case 2, 3, and 4, you're going to want to add 200 microliters of your dilutant, which is DI water, first. After adding that dilutant, you're going to take 200 microliters from the previous well and add it into your new well. So for example, I would take 200 microliters out of the first well and then add that into the second well. I would mix inside the well and repeat, where I'm taking 200 microliters out of the second well and putting that into the third well, and so forth. Except at well 4, instead of I'd still take the 200 microliters out, but then I would just throw it away afterwards. Each partner is going to do this twice, so you should end up using all of these wells that I'm marking here. In addition, to give you guys some reference points, we also want you to pipette your acid and base solutions into an additional 4 wells each. After you finish generating these dilutions, your TA will then put it into the plate reader and will read out absorbances for you guys. After you finish this part of the lab, you're going to be counting some beads using the hemocytometer. Your TA will have a solution prepared for you, and you're just going to want to follow the instructions and pipette 100 microliters of that solution into your own 500 microliter aliquot of water. You're going to be using these beads for the rest of your time in 167L, so just want to make sure that you keep them safe and keep them in a place where you know where you can find them. One thing I do want to emphasize here is that whenever you're pipetting beads or cells or any solutions containing solids, make sure that the solution is well mixed prior to pipetting and removing it. If you don't, you're actually going to end up getting a not very well mixed solution, and it won't be very repeatable. Your TA will emphasize this in section as well, so please do so. One last thing before we close up for today. is that it's important for you to understand this calculation here and why multiplying by 10 to the fourth gives you the number of particles per mil. This particular problem will appear in future tests, I can guarantee it, so please make sure to memorize this. That's all for today. As always, if you have any questions, please let me know via email